A very good day to Professor Dr. Edmund and Dr. Leslie. Today, my group, Group 1, Subgroup 2, will be presenting to you about malaria. My team members consisting of 5 members. They are Nurul Alia, Rebecca Shireen, me, Petrus Luna, Daeng Nur Hidayah, and Nur Shafika.
Malaria is a deadly disease caused by Plasmodium parasites spread by infected female Anopheles mosquitoes. It is most severe in sub-Saharan Africa with over 90% of global death. P. falciparum, the most dangerous species, is common there, while P. vivax is found elsewhere. In 2023, there were 263 million cases and 597,000 deaths worldwide. Symptoms appear 10 to 15 days after infection and range from fever and chills to seizures and breathing problems. High-risk groups include young children, pregnant women, people with HIV, and travelers. If untreated, P. falciparum can be fatal within 24 hours. Despite current tools, resistance to drugs and insecticide is growing, highlighting the urgent need for new biotechnological solutions.
As for the biological pathway, it is divided into two, which are the transmission and the liver stage. So malaria is transmitted to human through the bite of an infected female Anopheles mosquito, which is primary vector of the disease. Only female mosquito bite human because they need blood for egg production. During the blood meal, if the mosquito is carrying the Plasmodium parasite, usually Plasmodium falciparum, vivax, ovale, or malariae, it injects sporozoites into the bloodstream via its saliva. And then for the sporozoite injection, the sporozoites are the infective stage of the parasite. So these motile sporozoites rapidly travel through the blood and reach the liver within only 30 minutes post-infection. So this is a crucial step because the parasite must reach the liver to multiply and progress through its life cycle. Next will be the liver stage. So once in the liver, the sporozoite will invade the hepatocyte, which is the liver cell. Inside each hepatocyte, the sporozoites undergo a sexual reproduction, a process called exoitrostatic schizogony. They multiply massively, forming thousands of merozoites within a single affected liver cell. After several days, typically 7 to 10 days depending on the species, the hepatocyte will burst and releasing the merozoites into the bloodstream. So these merozoites then initiate the blood stage of infection by invading red blood cell, which is the erythrocytes, and this blood stage is responsible for clinical symptoms of malaria such as fever, chills, and anemia.
When a person is bitten by an infected Anopheles mosquito, the malaria parasite enters the bloodstream and heads straight to the liver. So why the liver? Because it offers the perfect environment, rich in nutrients and low in immune defenses, allowing the parasite to multiply suddenly before showing symptoms. This hidden liver stage is a crucial launchpad, producing thousands of merozoites that flood the bloodstream. But the real culprit behind the malaria spread is the Anopheles mosquito. These mosquitoes are expert vectors. They prefer feeding on humans, bite mainly at night when people are vulnerable, and live long enough to allow the parasite to complete its development inside them. They breed near human settlements, increasing exposure risk. Their salivary glands even serve as a safe storage site for the parasites. All these traits, human preferences, stealthy nighttime biting, and strategic biology make Anopheles mosquito dangerously efficient at spreading malaria.
Next is overview of gene drive technology. At the heart of modern gene drive system is the CRISPR-Cas9 genome editing tool. CRISPR-Cas9 acts like molecular scissors, allowing scientists to precisely cut DNA at specific location and insert, delete, or alter genes. In a gene drive, CRISPR-Cas9 is engineered into the genome of an organism such as a mosquito, so that when it mates, the gene drive copies itself onto the corresponding chromosome in the offspring. This process ensures that nearly all progeny inherit the modified gene, rather than the usual 50% inheritance rate seen with standard Mendelian genetics. Gene drive technology has been applied to mosquito, the primary vectors of malaria, to help combat the disease. Scientists can use CRISPR-Cas9 to introduce genes that either render mosquitoes resistant to the malaria parasite or reduce their ability to reproduce. For example, a gene drive might be designed to spread a trait that makes female mosquitoes sterile or unable to transmit Plasmodium parasites. By releasing a relatively small number of genetically modified mosquitoes into the wild, the gene drive can quickly spread the desired trait throughout the mosquito population. This approach has the potential to significantly reduce malaria transmission and ultimately the incidence of the disease. A case study by Regent et al. 2017 demonstrated that genetically modifying disease-carrying mosquitoes can control vector-borne disease. By using genome editing tools, specifically ZFN, TALEN, and CRISPR-Cas9, scientists can either disable critical genes or insert harmful genes in mosquitoes. These targeted genetic changes disrupt the mosquito's ability to transmit diseases, providing a strategy to reduce infection rates. Another report by Hall et al. 2015 demonstrated that using CRISPR-Cas9 gene editing to disable the male-determining next gene in genetically male mosquitoes, resulting in fully feminized development. Furthermore, this study showed that artificially activating the next gene in genetically female mosquitoes trigger male development. This confirms that next alone can initiate male traits, providing a pathway to convert disease-transmitting female mosquitoes into harmless males.
Despite its potential, gene drive technology poses significant ethical and ecological risks. It And then by Oye et al. 2014, its irreversible impact on ecosystem is a major concern with unknown long-term effects and possible harm to non-target species. And then also ethical issues also arise around community concern as affected population may lack input in deployment decisions, underscoring the need for transparent engagement. And this was done by World Health Organization in 2021. Additionally, unintended spread or misuse gene drive highlights the need for strong international regulation and biosafety control. And then in 2024, a research was done by World Health Organization that a key application of gene drive is controlling mosquito population by inducing female sterility or blocking malaria transmission, thereby reducing the disease incidence. This method offers a cost-effective, sustainable alternative to conventional vector control as gene drive require minimal release and self-propagate, reducing long-term public health intervention needs. This was said by S. Veld et al. 2014.
And that is all from us. Thank you very much for listening.